Mechanism of enzyme action. 1. What is the enzyme-substrate complex? The temporary binding of a substrate to the enzyme's active site forms the enzyme-substrate S complex. 2. What happens after the enzyme-substrate complex is formed? The substrate is converted into the product while attached to the enzyme, forming an enzyme product A complex. 3. What happens to the enzyme after releasing the product? The enzyme remains unchanged and is ready to catalyze another reaction. Models of enzyme action. 4. Who proposed the lock and key model, and when? Emil Fischer proposed the lock and key model in 1894. 5. What does the lock and key model explain? It explains that the substrate fits into the enzyme's active site like a key into a lock due to complementary shapes. 6. Why are enzymes highly specific according to the lock and key model? Because the active site's shape only allows a specific substrate to bind. 7. What are non-regulatory enzymes? Enzymes that follow the lock and key model, such as secrase and maltase, are called non-regulatory enzymes. 8. What is absolute specificity in enzymes? When an enzyme catalyzes only one specific substrate, e.g., urease catalyzing urea. 9. Who proposed the induced fit model, and when? Daniel Koshland proposed the induced fit model in 1959. 10. How does the induced fit model differ from the lock and key model? In the induced fit model, the active site is flexible and changes shape to better fit the substrate. 11. What is an example of an enzyme that follows the induced fit model? Hexakinase. 12. What are regulatory enzymes? Enzymes that follow the induced fit model and can catalyze multiple related reactions, e.g., carbonic anhydrase. 13. How does carbonic anhydrase function? It facilitates the addition of oxygen to hemoglobin and controls the formation of carbonic acid and bicarbonates. 14. What is the key difference between the two models of enzyme action? The lock and key model describes a rigid active site, while the induced fit model describes a flexible active site. Energy of activation. 15. What is activation energy? The energy required to cause molecules to react with one another. 16. Why can't living systems use heat as activation energy? Because heat would denature the temperature-sensitive proteins in living cells. 17. How do enzymes reduce activation energy? Enzymes lower the activation energy by forming a transitional complex with reactants. 18. What is a transitional complex? A temporary complex formed between an enzyme and reactants during a reaction. 19. Why are enzymes important in living systems? They enable reactions to occur under isothermal conditions without the need for high temperatures. Factors affecting the rate of enzymatic action. 20. What is the rate of enzymatic reaction? It is the amount of substrate converted or product formed per unit time. Temperature. 21. How does temperature affect enzymatic reactions? Increasing temperature speeds up the reaction until it reaches the optimum temperature. 22. What is the optimum temperature for human enzymes? Approximately 37 to 38 degrees Celsius. 23. What happens to enzymes above their optimum temperature? They denature, losing their specific structure and activity. 24. What is denaturation? The disruption of an enzyme's secondary and tertiary structure due to high temperatures. 25. What happens to enzymes at freezing temperatures? They are inactivated but not denatured and regain activity when warmed. 26. What is the minimum temperature for enzyme reactivation? The temperature at which an inactive enzyme becomes active again. 27. Why can some bacterial enzymes function at high temperatures? They are adapted to environments like hot springs and have high optimum temperatures. 28. What is an example of a high-temperature enzyme used commercially? Enzymes in biological washing powders for high-temperature washes. pH. 29. What is optimum pH? The pH range at which an enzyme shows maximum activity. 30. How does pH affect enzyme conformation? pH influences the ionic charges on amino acids, affecting enzyme structure and function. 31. What happens to enzymes in extreme pH conditions? They denature due to disrupted ionic bonding. 32. What is the optimum pH range for most enzymes? Between pH 6 to 8. 33. Give examples of enzymes with specific pH requirements. Pepsin, active at pH 2, acidic medium. Trypsin, active at pH 8, alkaline medium. 34. What is papain? A protease enzyme from papaya that works in both acidic and alkaline conditions. Enzyme concentration. 35. How does enzyme concentration affect reaction rate? Increasing enzyme concentration increases the reaction rate until equilibrium is reached. 36. What happens at equilibrium? The reaction rate plateaus as all substrates are bound to enzymes. 37. Why does increasing enzyme concentration stop affecting the reaction rate after a certain point? Because all substrates are already being catalyzed by the available enzymes. 38. What is the role of active sites in enzymatic reactions? Active sites bind to substrates, enabling their conversion into products. Thanks for watching.